areas in non-tumored animals where the disease doesn't occur, for reasons we're not sure, there's no difference in the immune status in these animals. So we know that once they get these tumors, their immune system starts going south. Also a really good example, and I'll come back on this with corals, that the concept of opportunism, and you hear that bantied about a lot in some ecology literature about opportunistic diseases. But it's really crucial when you banty that term about that you also link it with the immune status of the animal and the immune function. A lot of people just say, oh, well, this animal has got opportunistic disease. Well, opportunism is really that your immune system fails, and then you get infectious agents that come in. Just like AIDS. AIDS patients have opportunistic diseases because the virus is killing their immune cells, and then they're getting things like tuberculosis or cryptococcal meningitis or all those fun things, right? But they've made that link because they were able to show sequentially that it's the immune system that shuts down and then the bacteria or the pathogens come in. So again, just another thing. They also have these, a lot of these turtles have these vascular trematodes, these parasites that live in their blood vessels that cause all sorts of inflammation. We don't know the life cycle of these parasites. Interestingly enough, we know that most of these turtles probably get their parasites near shore and not in the pelagic environment. Now, how did we figure this out? Because remember I told you, we don't know what's happening pelagically with these turtles. Well, the way we figured this out is because we have a longline fishery, and the longline fishery is supposed to report all bite-caught sea turtles, collect them on the ships, and bring them back to Honolulu for necropsy. So this is another thing you do, is you try to partner up with agencies to try to opportunistically try to get samples to understand the different health status of the life history. And so this illustrates too that when you're looking at these marine animals, the health of these marine animals near shore is very different than the health in the pelagic environment. So you're looking really two different life history stages. And so you're trying to understand disease in these animal populations. You have to take into account what's happening pelagically and near shore. And then finally, we're finding things like it doesn't take, one turtle can transmit the disease to many animals. So there's this phenomenon called super spreaders. And they're actually finding this a lot now with a lot of infectious disease. When you look at things like AIDS, most of the AIDS cases are caused by just a few individuals moving that virus around to a lot of different people, rather than just a big sea of sick people transmitting it to a big sea of other susceptible animals. So this is being examined a lot more in animal populations. And we're also finding too that the smaller tumors tend to shed a lot more virus than the bigger tumors, and we can determine that from histopathology. And that's useful from a research standpoint, because then that helps you target tumors for, if you want to try to isolate the virus, you're probably going to try to use smaller tumors because they tend to shed more virus. So that can be helpful from a research standpoint. Okay, that's it for turtles. Any questions? Yes?
So the initial infection experiment in turtles.
Larry's?
Yeah,
Uh-huh.
I assume you couldn't do that now. I mean, I'm not suggesting we redo it because it's been done, but like
you could do it with justification. For example, if you were able to grow a virus, I always made the statement: if I can grow the virus in culture, I will go to DC to try to get a permit to make this happen, experimental infection. But otherwise, it would be really difficult to justify.
Okay, I was just curious because I think that's one of the things that always
there's no
makes
animal.
it harder for us.
There's no animal model. You're absolutely right. There's no animal, and this is another challenge. A lot of these a lot of these diseases, when you look at them, they have model systems. Like human diseases, people use white mice. In cattle, you use cows. In dogs, people use dogs for models. But we just don't have models for a lot of these animals, and that's a huge limitation. I totally agree. Okay, so fish. One of the big reasons why we don't do a lot of fish disease is this. When a fish gets sick, they usually get eaten. So you don't really have much of a chance, unless it's a huge mortality event that's really acute, you're not going to get really good demographic data on fish diseases. We had an interesting event, though, in 2010, where we had these guys. They're called the stripe-belly pufferfish. And we had reports of these fish basically attacking each other on the reef and causing these really gnarly skin lesions I mean, what these guys do is they're corallivores, and they have these huge teeth that can basically bite through anything. And they were biting each other, they were attacking each other, they were even attacking divers. And they were showing up, floating, puffed up on the surface. When a pufferfish is really stressed, they puff up. And we saw a lot of these things over about several months period, all throughout the Hawaiian archipelago. So between like June and October, we had this big event where these fish were popping up, floating, or these divers were seeing these fish attacking each other. And I think the only reason that we got so many fish to look at, we ended up looking at like 120 fish or something like that. Anybody have any idea why there's so many fish available to look at? Anybody know anything about puffers?
You don't want to eat
They're
them.
toxic. That's exactly right. They're toxic. And they've got this toxin called tetrodotoxin. And I think the only reason we were able to get a good sample size is that nothing was going to eat these things, because these things were totally, we have sharks and jacks all over the place in the ocean. I mean, if you're sick, you're going to get nailed, and these things were not getting touched at all. So that was actually very cool, because it gave us some good numbers to look at. And it turned out that on gross pathology, the major lesions we saw were these really hyperinflated swim bladders and these really enlarged discolored livers. And when we looked at the microscopy, these livers look very typically like toxic livers, almost like an alcoholic. I mean, if you see an alcoholic who's really chronically alcoholic, their livers will look just like that. They're full of vacuoles because the cells are all swelling and bursting. The gills were all falling apart, and then they had these really weird, this is the normal red cell of a puffer. It's kind of oval with the nucleus. And they had these really odd-shaped red cells that looked, for all practical purposes, like a toxic type response. So we did all the assays, we did the bacteriology, we did the virology, we did all the things to try to rule out infectious agents. And we were really backed in the corner that these things, the pattern, the widespread pattern didn't suggest a point source pollution, right? So we had fish dying all over these different islands. They had lesions that were very suggestive of toxicosis. We didn't have any man-made poison to point to as to what could be causing this. So our ultimate conclusion was probably some sort of marine toxin. Well, this is another huge, big hole that we have in marine diseases. There's a lot of toxins out there that we don't know anything about. And actually characterizing novel marine toxins is really, really hard. And we've been working for 5 years now with a marine chemist. And it's a lot of chromatography and fractionation and extracting organs. And it's really difficult discovering new toxins. And so this is a field that's really ripe to be turned on its head to try to figure out a more efficient way to do that. Because there's many cases we've seen in birds and other animals where we think it's been a natural marine toxin, but we just can't pin down the toxin because it's so labor-intensive and laborious to figure that out. So you can see as we're going down, we're learning less and less, yeah?
With these things occurring naturally sometimes, how do you approach second investigations as opposed to first investigations? Is there a fundamental difference in how you approach that?
So if this were to happen again?
Yeah, or something that if you came to a point where you said this is about the limit of where we can go with this, we think it's this or that or the other thing, you don't pursue it any further at that point, but then it pops up again in a new location or a third time. How do you restart that process?
Well, we go back, we reconfirm the same type of lesion, first of all. If we get the same type of lesion, then we go directly, this first time around, we were working with virologists, working with marine, we were working with a variety of different people trying to cast our net. This time I would go directly to marine biochemists. I would say, this is the data that we've gotten up to this point. This is the toxin that we've crystallized. We think it's, right now I think we think it's a glycoside of some sort, but we haven't gotten the actual molecular formula. And I would say, we've gotten to this point now, here's all the data, you need to move it forward to the end point. And that's basically how it works. So it is a kind of a recursive process, but it's not a total waste of time because you have this body of information you can build upon. Urchins, we had a die-off of urchins. These are collector urchins. Urchins are really important in coral reef ecosystems. Can anybody tell me why? Right, they eat algae, right? Remember coral reefs, it's a big battle between fast-growing algae and slower-growing corals. In fact, the reason you guys have lost most of your reefs out here, it all started with an urchin die-off in the 1980s. And we had this die-off of collector urchins in last year, actually, and they were showing up with these clinical signs. And again, we did histopathology in these guys, and they had these really funky, large cells. And when we did electron microscopy, we found these structures here that look like they're growing inside the cells. And for all practical purposes, we think it's some sort of virus, but we cannot put our hands on it. We've sent it to USAMRID. They've done their whole molecular stuff. They haven't found anything. We've sent it to people who do RNA, where they look, you can do the RNA of the host. They haven't found anything. And so this is the thing, is that the molecular, if you don't have the primers, it's really hard sometimes. So, we're kind of sucking air on this one. We
love to sequence that, or to try.
Yeah.
We might fail.
Yeah.
But
Yeah.
I'll give it to Liz.
Yeah, we thought initially it could be a plant virus, because there's some plant virus that like filiform like that. But the point of this is sometimes you'll see some of these disease investigations, people will go right to molecular, and they'll go right to molecular and say, oh, this is it because of the molecular, but they don't really relate it back to what's happening at the cellular level. And I have a big problem with that. At least here we have something to chase. We have something that's tubular, so we can at least, there's certain types of viral morphology that are tubular, others are not. So it gives us something to hang our hat on. So even though we may not have the answer, it's something to chase. And then there's coral reefs, and that's my big bugaboo, because coral reefs has been really the poster child in my book of the wrong way to go about coral disease investigations. I mean, you've had people, inferring causation based on gross lesions, picking their own etiology, using molecular tools to incriminate infectious agents with no basis on the cellular level. So it's been really, and because there's 40 years of literature on this, you're really swimming against the tide, trying to say, hey, folks, there's a better way to approach this than spinning your wheels. So we're basically trying to do things a little bit differently in the Pacific, where most of our coral resources exist, by the way. And so we're doing just like what we do in wildlife. We look at things at the cellular level, and we're finding really cool stuff. We're finding fungi killing corals. We're finding helminths. We're finding parasitic corals. We're finding all sorts of really interesting things. But it's really hard to get traction in the coral disease community because they're all focused on Vibrios. You think Vibrios are killing corals, or this bacteria is killing corals, with absolutely just no evidence at the helminth or worms. Worms, yeah, it's okay. Yeah, so I mean, there's lots of cool stuff out there that we're seeing that needs to be looked at. It's just trying to get traction on it. But just to show you the value of this approach, a recent example, we had a big die-off on North Kauai of corals, affecting a variety of corals. So you can see this is normal coral right here. This is tissue loss. And then this is normal coral over here in a big patch of tissue loss here, tissue loss there. So we did our investigation, and within a month we had an idea of what was killing this coral. So we knew, we did the microbiology, we did the pathology, and we saw cyanobacteria associated with cell death. And so we said, you have an epizootic here. You have an epidemic disease of cyanobacteria killing these corals. So the manager goes, well, how do you know it's an epizootic? How do you know it's a cyanobacteria? Well, I know it's a cyanobacteria because here it is. I've got photographs of it killing the cells on histology. And they go, well, how do you know it's an epizootic? And I go, I know it's an epizootic, which is an epidemic for animals, because we've been looking at corals since 2001. So when you do this histopathology, what you do is you take a piece of tissue, you put it in formalin, and then you put it in a block of paraffin. You slice it up, you put it on a microscope slide, and you look at it on the microscope. But that block of paraffin will last forever. And so that's an archive. And so you have a library of tissues that you keep. We keep all these paraffin blocks. And so I was able to go back to my archive tissues and say, I have looked at this number of corals over the last 10 years. And these are the number of cases of cyanobacteria that I've seen. And here's where we are right now in 2012. So in some ways, we were way ahead of the curve compared to the Caribbean. I mean, in the Caribbean, they're saying, oh, we have coral mortalities, we know what's killing the corals. They don't even have that archival tissue bank to go back to say whether, if you have a new phenomenon in your population, is it something really new? Or is it something that was at low levels in the population and it's flaring up? And from a management standpoint, that can be huge. So that was really an eye-opener for me, the value of this, having this tool. And then there's really some cool stuff you can find out just biologically using, looking at coral cells. So, corals are relatively simple animals. They've got an epidermis, they've got a connective tissue layer, they've got a cell layer called a gastrodermis with a symbiotic algae, and then they've got a stomach, and then they've got these cells that secrete the skeleton. And these symbiotic algae are called zooxanthellae. This is what they look like on the tissue. And when they bleach, that's when the algae leave the tissue. And so what you have is you have this transparent tissue over a white skeleton, which makes the corals bleach. So the zooxanthellae give color to the coral. Well, we found, so that's a big symbiosis. In fact, zooxanthellae were discovered in Palau by the Palau Marine Biological Institute, Japanese biologists. They're the ones who discovered it in like the early 1930s. And it's been a very classical example of animal-plant symbioses. And a lot of work has been done on coral algal symbioses.
Well, it actually goes back a lot farther than that.
Really?
The Germans, yeah.
Really?
Much farther than that.
Oh, cool. I didn't know that. Okay. Yeah, because I just knew about the Palau Biological Institute. Well, it turns out that corals not only have symbiotic algae, they have symbiotic bacteria. So you find in normal tissues, you'll find these microcolonies of bacteria either in the epidermis or the mesenteric filaments. Perfectly happy, no pathology, perfectly normal animals. And interestingly enough, you find that these bacterial colonies are upregulated when the colony has got lesions on it. So it's almost like they use them as an immune response. And when you look at the biogeographic pattern of these things, you find that there's a lot more of them near the coral triangle, and as you radiate away from the coral triangle, there's fewer and fewer of them. So it looks like they seem to have originated somewhere in the coral triangle. But most interestingly of all, I think, is that they're most prevalent in the dominant species of dominant genera of corals. So when you go to a reef, when you jump on a reef, and you look at the corals, most of those corals are going to be a particular genus. And that reef is usually going to be dominated by Porites, Passalopora, or Acropora. And when you look at all the different genera of corals, we have a big bank now of like 113 species of corals with like 60 genera. The majority of the genera that have these bacterial symbionts are these three genera. So I'm thinking that they must be providing some competitive advantage. And we know this from insects. I mean, insects have symbiotic bacterial colonies in their tissues that are crucial for things like, evolution, immunity, species assemblage. So there's no reason why corals can be doing the same thing. So I mean, this is a whole huge avenue, I think, of investigation that nobody's really paid attention to,
Not true.
Really? I
published extensively on this. Many other people have.
On these cell-associated bacterial
Not
aggregates?
on bacteria, but
No.
on zooxanthellae.
Oh no, zooxanthellae.
Competitive advantage, yeah.
Right. No, I'm not saying that. Totally, I agree.
There are some papers out about the bacteria.
Right.
Most of them are mucus focused,
Right.
as opposed to the cellular,
Yeah, yeah.
right?
Right. No, I totally agree with you. The zooxanthellae, there's a huge body of literature. What I'm saying is that people have been overlooking something that could be almost as equally important, at least for these particular genera. And I don't know how we can culture these things, but this is another thing we could collaborate on. So this is a good example of the value of looking at the animal cells to give you a more holistic picture of what these things are doing. And that's pretty much all I have.
I can speak to the coral problem in Florida, having been diving and photographing them since about in the 1950s, and watched the whole thing develop. And when it became very clear that we had coral diseases, starting in the late 70s, but peaking in 1983, nobody wanted to do this kind of work. I dealt with a marine sanctuary down here, and they were too busy worrying about anchor damage and simple things like that. but you couldn't get anyone. It took at least 15 years, I think, for people got interested and finally started looking for microbes and diseases in the corals. Now, you guys had I guess you didn't really get them until much later, and I guess you started around 2000 or something like that.
Well, we've been looking since 2000. You know, I can't really speak for before then because I wasn't really focusing on corals at the time. But, I mean, the reason I got involved in it was because 95% of U.S. coral reef resources under U.S. jurisdiction are in the Pacific. You know, we have all these refuges, these island refuges, with all these coral reef resources. We can't afford to repeat what you guys did here. We simply can't do it, you know. So we're trying to figure out some other way to, you know, be ahead of the curve. Because, I mean, this is a disaster what's happening here. I mean, it's horrible.
Oh, absolutely.
I mean, you read, I just read on the papers. You know, I mean, I went diving with Mark, actually. We went to the Florida Keys, and where they're growing these acroporas on trees to receive them, and those look pathetic. They had algae all over them, and it was very sad to see, you know, what's happening.
That peaked in 1983, the same time the diademas died off
Yeah,
over the Caribbean.
and nobody looked into diademas either.
No. It took a while. It took a long time. But right now, there's not enough diademas
Right.
to work on.
Yeah. Yeah. Yeah.
Would you mind going back to the pufferfish, the distribution slide?
Sure. So the whites is where we got reports, and the blues are where we actually got specimens
Okay.
for necropsy.
And that was in the summer of, what was it?
2010.
2010.
Yeah, if you have any bright ideas, let me know.
Did you ever check the phytoplankton for the
So
toxin?
I looked, I asked people about phytoplankton blooms. Typically when we look at these marine toxins, we look at the stomach contents because that's when we get the clues, right? I mean, because typically they have, like when we found domoic acid in pelicans in California, they all had anchovies inside their stomachs. So it was really, we could make the connection that the anchovies were grazing the pseudonychia and bioaccumulating. The problem with these guys is they're basically omnivores. So some had crustacea, some had corals, some had coral and algae. You know, also it was a whole gamut of different things. So we really didn't have a common stomach content source to go for. So it was really hard. Yeah.
Now, you do get plenty of Asian dust in the Pacific. Right? In fact, I think the red soils that grow the pineapples on is actually Asian dust. And of course, we get African dust. So we jumped on the African dust, and we got a whole program at USGS for quite a while.
Yeah, I'm skeptical about that whole theory, though, honestly. Yeah.